Good afternoon, one and all. I am Dr. Srijani Banerjee, Assistant Professor of Department of Biotechnology, School of Life Science and Biotechnology, Adams University, Kolkata, India. For today's renewable energy session, I am going to present my PhD work, which was being carried out in Indian Institute of Technology, Kharagpur, under the supervision of Professor Debabrata Das from Department of Biotechnology and Dr. Arnav Atta from Department of Chemical Engineering. So the topic of my today's talk is process parameter optimization for microalgal biodiesel production in a biorefinery approach. The demand for renewable energy sources is increasing day by day, mainly because of the huge consumption of fossil fuel which have limited reserve. Development of energy sustainability is essential for overcoming the energy crisis of the world. The alarming increase of the greenhouse gases in the atmosphere is significantly affecting global warming problem. So the present world is searching for some alternative source of energy which can mitigate the global warming problem and as well as meet the global energy demand. So the global energy scenario depicts that by 2050, the world energy demand will exponentially increase. The fossil fuels are non-renewable sources, they are finite source of energy and it emits environmental pollution or it emits greenhouse gases to the atmosphere. So the attention has been shifted towards the alternative source of energy which are renewable, eco-friendly and sustainable. So till date, there are mostly three generations of biomass-based feedstock. The first one is the first generation biofuel feedstock, that is the agricultural feedstock. There comes the food versus fuel dilemma because agricultural feed requires larger amount of land to cultivate and it also has the higher growth time. Then to mitigate the drawbacks of the first generation biofuel comes the second generation biofuel, that is the lignocellulosic feedstock based. There comes the difficulty in pre-treatment for the biofuel extraction from the lignocellulosic feedstock and it also has the longer growth time. To mitigate the problem of both first and second generation biomass based feedstock comes the third generation biofuel, that is the microalgal biomass as feedstock, which possesses faster growth rate than the normal terrestrial plants and it is easy to extract biofuel from the microalgae. So why microalgae? So microalgae possess higher photosynthetic rate than the normal terrestrial plants. It has also higher faster growth rate. It can sequester CO2 from the atmosphere. It reduces greenhouse the biofuel which are being extracted from microalgae reduces less particulate matter emission to the pollution. It is mostly sustainable and mostly no food versus fuel debate comes for microalgae cultivation as it requires no not larger lands to cultivate microalgae. So it can be grown in wastewater also. So the various application of algae can be used as food supplement because it is very much rich in protein content. Algae can be converted into biofertilizer. Algal feedstock is very much rich in lipid and carbohydrate content which can be further converted into biofuels like biodiesel, biohydrogen, bioethanol, biohydrogen and biogas. So why biodiesel from microalgae and what is biodiesel? So biodiesels are the monoalkyl esters of long chain fatty acids produced from transesterification reactions with the help of methanol and different types of catalyst. So the advantage of biodiesel is that it reduces emission of carbon monoxide, hydrocarbon and SOx emission. It has higher C10 index. It is safe to use and it has higher flash point and mostly it requires no engine modification to be used in the presently used automobile engine. So from the literature survey, it was being found out that Botrycoccus browni, Neochloris oleoabundans and Nanochloropsis are some of the microalgal strains which contain higher lipid, and it was being also found out some gaps that lower biomass concentration with higher lipid content can be a solution for the commercialization of biofuel or biodiesel. Scarcity of suitable design of photobioreactor for the proper cultivation of microalgae is there. Very few study on the correlation between the hydrodynamics parameter and biomass and lipid production in photobioreactor is there. Metabolic behavior and flux distribution study of the process related to biodiesel production is less explored, and very few literature on the impact of the microalgal biodiesel production process to the environment is available. So the objective was set for the work was selection of high lipid containing algal strains and suitable physicochemical parameter for the improvement of the biodiesel production, influence of hydrodynamic parameters of the photobioreactor on algal biomass and lipid production, to study the flux distribution. That is very important to find out the metabolic behavior of the organism, why the organism is producing higher amount of lipid. And the fourth one is development of suitable transesterification technique, because with the transesterification technique, the lipid will be converted into biodiesel. So that technique has to be optimized for biodiesel production. And finally, the overall life cycle assessment of the process. That means what will be the environmental impact of the process that was being studied. So these are the materials and methods used for the study. So the first objective is selection of high lipid containing algal strains and suitable physicochemical parameters for the improvement of the biodiesel production. So this work was being published in INDC journal by SE. So this is the work plan: selection of suitable algal strains for biodiesel production. So the strains which are being used for the present study is Chlorella soropiana, Chlamydomonas reinhardti, Anabaena species, Botryococcus brownie, Synodesmus, and Neochloris olivibundans Utex 11H. So from the biochemical analysis, it was found out that Botryococcus brownie has the higher lipid content, but that lipid is rich with hydrocarbons, large hydrocarbons that can be further converted into gasoline rather than diesel. But the Neochloris olivibundans has the lipid content of 29% that can be further converted into biodiesel, as it consists of fatty acid chain lengths from C16 to C18, that is the oleic acid, linoleic acid, palmitic acid. So that those are the most suitable one for converting into biodiesel. So and from the growth characteristics also, it was being found out that Botryococcus brownie has the doubling time of 4.44, that is very slow growing, whereas Neochloris olivibundans is relatively faster growing, as it has the doubling time of 2.52 days. So the media was optimized for the growth of the Neochloris olivibundans, and modified Bolthrian media was found out to be the suitable one for the growth of the Neochloris olivibundans. So the pH was being optimized, and it was being optimized as 6.5, as it gives the better biomass concentration of 1.21 gram per liter with a lipid content of 30% wet by weight. Then the temperature was optimized as 25 degrees centigrade, as it gives the higher biomass concentration with the higher lipid content. And also the activation energy was being calculated from the Arrhenius equation for this specific organism, that is 23.4 kilojoule per mole. Then the nitrate concentration in the media plays a pivotal role, because nitrogen is the rich source for the biomass production. So it was being optimized as 4.41 millimolar with the biomass concentration of 1.12 gram per liter. 8.81 millimolar, 82 millimolar gives the higher biomass concentration, but the lipid content is somewhat less. We all know that in the nitrate stress condition, the lipid content actually enhances. That's why to meet both the demand, we have chosen 4.41 millimolar nitrate concentration, as it gives the lipid content better than 8.82 millimolar. So multiparameter was being optimized by the Taguchi model. 9-factor Taguchi model was being actually used, and the MITAB software package, the statistical software package, was being used. This is the design matrix for 3-factor Taguchi model, and response table gives that the temperature is the most influential parameter for the growth of this specific microalgae, and 25 degrees centigrade temperature with pH 7 and 4.41 millimolar nitrate concentration gives the higher biomass concentration of 1.550 gram per liter with the lipid concentration of 0.625 gram per liter. So with the optimized parameter, the cultivation was being carried out in 3 different types of photobioreactor, that is airlift, bubble column, and flat panel, and it was being found out that bubble column gives the higher biomass concentration with higher lipid content and with the doubling time of 2.24 days. So why in the bubble column reactor the biomass is actually coming high? To find out the reason, a basic CFD study was being carried out, and it was being observed from the CFD study result that the dead zone is getting minimized as the homogenized condition is achieved inside the bubble column reactor. As we can see that these blue spots are the dead zones. So we can see that the dead zones are getting minimized inside the bubble column reactor. This is the reason for better biomass production inside the bubble column reactor. Lipid accumulation study was done by the Nile staining technique and observing it under the confocal microscopy, and these bright yellow spots are the lipid droplets inside the microalgae, and it was being observed that with the cultivation time enhances, the accumulation is also getting enhanced. The CO2 sequestration study was carried out by sequestering different concentration of CO2 into the microalgal cultivation inside the reactor, and 2.5% volume by volume CO2 air mixture gives the better biomass concentration of 2.56 gram per liter with the lipid content of 49% by weight, and the elemental analysis gives that 1.503 gram of CO2 is being sequestered by 1 gram of algal biomass, or we can say it, for producing 1 gram of algal biomass, 1.503 gram of CO2 is being sequestered from the atmosphere. And the FAME analysis was finally being done by the gas chromatographic analysis, GCFID, and it gives that saturated to unsaturated, unsaturated to saturated fatty acid ratio is near to 70 to 30, which is the theoretical one, and the major fatty acids were obtained as palmitic acid, palmitoleic acid, oleic acid, and linoleic acid, which are suitable for biodiesel production. So the second objective was the influence of hydrodynamics parameter of the photobioreactor on algal biomass and lipid production. Although laboratory cultures of microalgae are ubiquitous, its large-scale cultivation is still restricted by high production cost and low yield. And what will be the interrelation between these hydrodynamics parameter like volumetric mass transfer coefficient, mixing time, reactor design, flow rate, shear stress effect are affecting the biomass concentration and lipid content, that was actually being studied in this objective. This work was being published in Bioprocess and Biosystem Engineering. So these are the schematics of the photobioreactors which were being taken consideration for the study. Airlift reactor with orifice purger, bubble column reactor with orifice purger, airlift reactor with ring purger, and bubble column reactor with ring purger. So three different types of parameter were chosen mostly, that is the flow rate, mass transfer, volumetric mass transfer coefficient, mixing time, and with the flow rate, the shear stress effect, how that is affecting the growth actually. So the mass transfer was being carried out by dynamic gassing out method, mixing time was being analyzed by acid tracer method, and shear stress was being analyzed by the viscometer and the rheometer. So the results gives that bubble column reactor with ring purger, by maintaining 600 ml per minute flow rate, gives the mixing time of 33 seconds, shear stress of 0.0419 Pascal, and volumetric mass transfer coefficient of 27.7 to R inverse gives the higher biomass concentration of 2.332 and with the lipid content of 49% weight by weight. Although we can see from the table that 800 ml per minute flow rate in the bubble column reactor ring purger gives the lesser mixing time, that is 30 seconds, and the higher volumetric mass transfer coefficient, that is 30.12, but it has the higher shear stress, which is actually affecting the biomass production inside the photobioreactor, because after cultivation of 2 days all the cell is getting died. So to reinforce the result, microscopic study was being done with the biomass collected from the reactor, which was being run under 800 ml per minute flow rate, and it was being observed from the picture that the shear stress effect is actually rupturing all the cells because it can't withstand that much of shear stress for cultivation of that microalgae. So for the further study, 600 ml per minute flow rate is considered for the study, as it is not causing any cell damage. So to again reconfirm the result, computational fluid dynamic study was being conducted. So CFD model based on Euler-Euler approach is developed for mostly bubble column reactor, as we have got a better result in bubble column reactor ring purger. So Reynolds number was first calculated, because we have to consider the model based on whether it is in laminar or in turbulent regime. So Euler-Euler multiphase model with implicit volume fraction formulation and K omega turbulence model are adopted. So Euler-Euler equation are solved by 3D bubble column reactor using a finite volume method based solver, and ANSYS Fluent 15 was being used. Flat velocity profile is implemented. Outlet is kept at atmospheric pressure. No slip boundary condition is implemented. Fine unstructured tetrahedral elements are selected as the meshing scheme. Meshing is of orthogonal quality. Standard K omega model is used. For turbulent interaction, Sato model is selected. And it was being found out from the result that air velocity, means this is basically the plot of air velocity distribution inside the photobioreactor at a specific plane, that is 15 centimeter from the inlet of the photobioreactor with varying flow rate. It shows that homogeneity was improved with increasing the flow rate. Means this is basically the first one. The figure A is for 100 ml, then it is for 400 ml, figure B, figure C is for 600 ml, and figure 8 for 800 ml. So the first figure, that is the figure A, portrays that higher velocity towards the center in all cases, indicating the air being directed towards the center of the reactor. Figure A depicts that a parabolic trend is being, we can see a parabolic trend which can be ascribed to a very low flow rate that gradually changes with an increase in inlet gas velocity. Whereas from figure B, we are getting a different trend, but homogeneity was attained at a much lower flow time. Figure C and D demonstrate flatter profiles which were attributed to the higher flow rates and flow regime shifts towards turbulent flow. So it was also observed from the CFD study, CFD simulation study, that 600 ml per minute is considered for further algal cultivation process in the bubble column photobioreactor. So to compare the experimental result with the simulated mixing time, this table is being formed and the calculation was being done, and it was found out from the table that for 600 ml per minute flow rate, the experimental results gives 33 seconds as the mixing time, whereas the CFD study gives the 32 seconds. That is mostly means we got 3.65% deviation from experimental to simulated data, which ascertain the CFD model reliability. To summarize this, these experimental results indicate pivotal role of sparger type, as we are replacing orifice sparger to ring sparger. That actually is getting that is actually putting some impact on the biomass concentration. Newly customized bubble column reactor with ring sparger leads to maximum biomass and lipid production. Shear stress decreases with orifice is replaced by ring sparger. A predictive CFD model for bubble column hydrodynamics advocates the efficacy of 600 ml per minute airflow rate to provide a desirable mixing type, which also produces a higher biomass concentration. So the third objective is to find out the metabolic behavior for the microalgae. So this is the work plan. Mostly it was being done in the mixotrophic mode of cultivation. So two different types of carbon source, that is glucose and acetate, was first considered for the mixotrophic cultivation, and it was found out that glucose as compared to acetate is more suitable for the growth of neochloris abundance. Which gives the biomass concentration of 2.34 gram per liter after 7 days of cultivation by maintaining 2.5 gram per liter of glucose in the medium. So cell growth kinetics was also calculated from the Monod equation and Lineweaver-Burke equation, and it was found out that doubling time of 6.28 hours, which is comparatively very less than the autotrophic condition, and also the biomass concentration was also getting enhanced in a mixotrophic mode of cultivation. So why? By supplementing glucose, the doubling time is getting less, and we are getting higher amount of biomass concentration. To find out that, the metabolic flux analysis study was being carried out with the 13C labeling pattern, that is the most commonly used technique for MFA study, and the 13C labeling pattern of intracellular metabolites are analyzed by GCMS study. So direct interpretation of the data identifies active pathways in a network. So MFA was formulated. Isotope level atoms with distinct leveling distribution are incorporated in the structure of various metabolites, and Metabolic network connectivity and atom mapping information is used to infer the flux distribution and minimize the sum of squared deviation between predicted and measured mass distribution are calculated. So this is the culture procedure for 13C MFA study. First, the means mixture has been taken U13C, 113C and unlabeled glucose. Basically the unlabeled 13C labeled glucose, 1 carbon labeled glucose and unlabeled glucose with the ratio of 1:1:3 was being supplemented in the media for the cultivation of microalgae, and it was being cultivated in the shaking incubator. Sampling was being done, harvesting of the cell, hydrolysis of the cell, then the derivatization, and finally the GCMS analysis was being done. And the glucose uptake rate was 1.23 millimole per gram dry cell weight per hour, and mu max value was achieved as 0.034. And metabolic flux analysis tool which was being used for the present study is FluxBite version 0.1.5, and the glucose uptake rate and the growth rate were taken as the basis fluxes. So these are the basic assumptions were taken for the study, and this is basically the metabolic flux map which was which we got after the flux analysis study. And from the map it was being observed that 11.5% of the net carbon intake was channeled towards such synthesis, starch synthesis, and the flux into the acetyl CoA sink was 33% of carbon taken up, reflecting a high lipid synthesis flux, because acetyl CoA is the main precursor for fatty acid synthesis from a microalgae or lipid production pathway. And both the glyoxylate shunt and ED pathway were active for the present organism, with later carrying about 29% of total carbon intake. So to summarize this, that the majority of flux is going into the acetyl CoA sink, which is 33% of the total carbon taken up, reflecting a high lipid synthesis flux. So the third objective and the final objective is to optimize or develop a suitable transesterification technique by which the lipid has to be converted into diesel. For that, the objective was set as development of suitable transesterification technique for biodiesel production and life cycle assessment of the overall process, which this work was being published in Energy Conversion and Management Journal. So this is the basic plan. So first, the microalgal biomass harvesting and drying accounts for 90% of the cost of the biodiesel production from microalgae. So if that can be reduced, biodiesel production from microalgae can be commercialized. So here autoflocculation technique was actually chosen for the harvesting of the biomass by just only changing the pH of the cultivation solution. The microalgae can be harvested. So maximum flocculation efficiency of 94.5% at pH 13 within 13 minutes of time was achieved, and with the zeta potential change from the minus 16.4 millivolt to minus 2.13 millivolt, which signifies that the flock formation is taking place. So as we all know that microalgal lipid is intracellular, so we have to extract out that intracellular lipid. Means we have to actually facilitate that lipid to get extracted from the microalgal cell. So different pre-treatments like autoclave, ultrasolution and microwave irradiation was given in the autoflocculated wet biomass. We are actually not drying the biomass, and it was being observed that autoclave, basically that is the heat, high heat treatment, gives the better lipid content after giving that pre-treatment, which gives 56.5% weight by weight of lipid that was being extracted from the autoclave pre-treated with microalgal biomass. So we conventionally use acid and base catalyst for the transesterification reaction, but mostly when we are using in situ transesterification technique where we are using a wet biomass, this acid and base catalyst is a drawback to use, because acid catalyst can corrode the reactor wall and it is also not reusable, and if the free fatty acid content is more than 7%, saponification reaction occurs if we use base catalyst nanocatalyst, and it is also not reusable. So the people is searching to make it more greener, different types of using of different types of nanocatalyst for the transesterification reaction, as it is sustainable and reusable. So why nanocatalyst in the transesterification reaction? Because it has higher catalytic activity, large specific surface area, high resistance to saponification. So nanocatalyst, as per the best of our knowledge, this was the first report that was reported by us for synthesizing the nanocatalyst from hibiscus plant leaf. And iron oxide nanoparticle was synthesized from leaf extracts of hibiscus. Iron oxide particle was first calcined at 700 degrees centigrade, and the XRD, FESIM, and TAME image reinforced the result that successfully the catalyst was being synthesized, as the size also matches with the normal library of nanocatalyst of iron oxide. So the ingredients which are being present in the hibiscus extract are matches also with the functional groups that has to be present inside the oxide nanoparticle. For that, FTIR was being actually conducted, and we also compared different types of catalyst for this biodiesel yield, and it was being found out that by using Fe2O3 nanoparticle produces higher amount of biodiesel as compared to acid and base catalyst. 81% biodiesel yield was obtained by using this iron oxide nanoparticle as catalyst. So the process parameter was also optimized by by using this type of iron oxide nanocatalyst. Means what will be the transesterification reaction time, temperature, and what will be the iron oxide concentration. And it was found out from the study that 1% weight by weight iron oxide catalyst concentration with 65 degrees centigrade temperature and within 4-hour reaction time, the lipid can be converted into biodiesel, which gives 86.5% FEM content, or the biodiesel yield. And again, the fatty acid methyl profile was being analyzed by GCFID analysis. And finally, the extracted biodiesel was compared with the ASTM and conventional diesel fuel, and all the properties like kinematic viscosity, density, calorific value, acid value, iodine value, flash point, pour point, and Seaton number was comparable with the ASTM and diesel standards, which actually proves the feedstock to be used as a biodiesel. So the activation energy and the thermodynamics parameter for transesterification was also optimized, and the values of thermodynamics parameters, that is del G, that is a Gibbs free energy, del H, and del S, shows that the reaction was unspontaneous, endothermic, and enderogonic in nature. The Aspen Plus process simulation was also carried out to find out the suitability of the process for the industrial use, and as we actually matched the experimental result with the simulated one to find out whether this process can be implemented in an industry level, and it gives that the transesterification results in a 90% biodiesel yield. Maximum deviation of 3.5% from the experimental result of biodiesel yield was obtained from the Aspen Plus process simulation, which actually signifies the process suitability in an industrial level. And finally, the life cycle assessment of the study was being carried out by the Gabi 7 software package, and the things which were being optimized by this LCA study is the means huge global warming potential, eutrophication potential, and human toxicity potential, and eutrophication potential or ozone depletion potential. And it gives that by using the iron oxide nanocatalyst, the process is much more environment friendly than by using the HCl or base as catalyst. Then to enhance the sustainability of the process, a biorefinery approach was being developed. The lipid was first extracted from the microalgal biomass, and from the lipid extracted algal biomass, a carbohydrate and protein content was analyzed, and it was found out that carbohydrate content of de-oiled algal biomass, means first the lipid was being extracted and it was converted into biodiesel. Then from the leftover biomass, the carbohydrate content is 20.2%, which can be further converted by dark fermentation for biohydrogen production using acidogenic mixed consortia, and by co-culturing the biomass with Saccharomyces cerevisiae, it can be further converted into ethanol, which gives actually proves the biorefinery approach, and the bioethanol production of 0.612 gram per liter was achieved, and the biohydrogen production of 485 milliliter per ml per liter was actually obtained. So this is the schematic of the overall work. To conclude, biomass and lipid production were maximized with concentration concomitant CO2 sequestration, which makes the process more sustainable. Bubble column reactor is a suitable photobioreactor for the cultivation of microalgae. A predictive CFD model for bubble column hydrodynamics advocates the efficacy of the airflow rate to provide a desirable condition for higher biomass production. MFA study revealed that the lyxolic shunt and ED pathway were active, and most of the flux is channelized towards acetyl-CoA. Biological production using pre-treated wet biomass can reduce the major cost of drying. Plant extracted iron oxide catalysts act as a suitable alternative to acid and base catalysts for transesterification reaction, and life cycle analysis of the present lipid extraction and transesterification process reduces the environmental impacts with respect to global warming, eutrophication potential, human toxicity potential, and ozone depletion potential. So these are the future scope of the study. These are some of the reference. These are some of the publication from my work, and acknowledgement to my supervisor. And this is the group where I worked with in IIT Kharagpur. And thank you this Applied Energy Symposium organized by MIT and Harvard for giving me this opportunity to share my work in this great platform. Thank you. One and all. I am Dr. Srijani Banerjee, Assistant Professor of Department of Biotechnology, School of Life Science and Biotechnology, Adams University, Kolkata. For today's renewable energy session, I am going to present my PhD work, which was being carried out in Indian Institute of Technology, Kharagpur, under the supervision of Professor Debabrata Das from Department of Biotechnology and Dr. Arnav Atar, Department of Chemical Engineering. So the topic of my today's presentation is process parameter optimization for microalgal biodiesel production in a biorefinery approach.